Hello everyone, I welcome you all once again to my series of lecture, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today we are going to discuss about one of the optimization technique, that is the Plackett-Burman Design of optimization. So why I have taken this optimized technique separately, it is the reason is that it is one of the best optimization technique to perform for a fermentation media. So for us, if you see that once we are designing a fermentation media, our core objective is that what are all should be the constituents present in the composition present in that fermentation media and what concentration it should present, so that we can get a maximum yield of the product. This is the core objective we will keep in the mind while designing the fermentation media. So at the time what we have to do is we have to perform an optimization technique. So the best one is the Plackett-Burman design, easy to perform, and we can get the results in a very precise graphical manner, right? So let us first understand, before getting into the topic, a basic understanding about fermentation. So as everybody knows that fermentation is a metabolic or biochemical process in which what happens is that the microorganism utilizes the substrate which is present in the media, and through its biochemical pathway it forms the product which is of our pharmaceutical importance or an industrial importance one. So for example, if you see what happens is that the yeast Saccharomyces cerevisiae will take up the glucose under anaerobic condition. Through its biochemical pathway, glycolytic pathway, it forms the ethanol as a secondary metabolite, which is of our industrial importance. Like that, if you see any fermentation process, we can get two types of products. Either it may be a primary metabolite or a secondary metabolite. So primary metabolite, or example like glutamic acid, which is of pharmaceutical importance one, or as ethanol or antibiotics, which is the secondary metabolite of pharmaceutical importance one. So to get this product, the first core is the media. So the fermentation media is the core to get a product yield at a higher concentration. So if you see the upstream process of fermentation, two things should be run parallelly. One is the optimization of the fermentation media. Second is the strain improvement. If these two things is perfect, then our product yield will be very high. So if you see the fermentation media, so we design the fermentation media based on the requirement of the microorganism. So if you see the microorganism, whether it's a bacteria, yeast, fungi, actinomycetes, anything, it requires water, an energy source in the form of a carbon source, the nitrogen source, minerals, everything. But the quantity and the type of the source will generally vary from each fermentation process. So we need a detailed investigation of each fermentation process, so that what the microorganism needs for the formation of the product. So this depends upon a better knowledge about the biochemical pathway of the microorganism. So if you know a better biochemical pathway of the microorganism, we can understand which substrate should be supplied to get the desired product in a high quantity. And further we can easily optimize the fermentation media. So today we will more concentrate in this optimization technique, because others we have a separate aspects to understand. So if we see the optimization, so optimization is nothing but we are getting an exact level of the nutrients which is needed for the product formation, which is important in the biochemical pathway for the product formation. So to start with the optimization, we have to have a better, clear objective. What is our objective? So objective of optimization, whether it is the production of biomass or a product formation. If it is for biomass, then we have a different constituents to be optimized. If it is a product, then the production media of the fermentation, we have different constituents. So whatever it may be, but the optimization technique remains the same. So the optimization technique are of different types available, but each type has its own advantages and limitations. So if you see the different methods of optimization, the very old method is the classical method. So what we will do in this classical method is simply we keep particular variable constant Then we started changing, we keep all variables constant except a particular variable which is varied until an optimized product yield is reached. Then we will keep this optimized level as constant, then started changing the next variable. So finally we can end up with an optimized fermentation media. But the problem with this technique is that it is time consuming and costly in nature because we have to perform more number of experiments. Then the second technique is the factorial design, which requires less experiments. So we can do with a less experiments, we can optimize two variables, but the problem is that we have a limitation. So we can go up to 4 variables only. If we go more than 4 variables, the number of experiments will start increasing exponentially. So that's limitation is the factorial design is having. So a better version of the factorial design as such is the Plackett-Burman design that we are going to discuss elaborately in this lecture. So in that we are having 2 techniques. One is the Box-Wilson method and the Hendrix method. So each method we will understand in a better manner. Then the final one is the simplex optimization technique. So let us understand the Plackett-Burman design. So Plackett-Burman design has its unique advantage. So first we understand the advantage of the Plackett-Burman design so that we can get an easy idea how we can perform it. So if we see that if the number of variables is more than 5, then with least number of experiments we can optimize the thing with this Plackett-Burman design. That is one of the major advantage. And the second one is, out of these 5 variables, which variable is more important for product formation, which variable is less important for product formation can be identified with this technique. Then further support by Box-Wilson method and Hendrix method, we can optimize these variables, right? So how we can do it? This I have shown with a simple illustration. So let us consider 7 variables we are having in a particular fermentation media, like a carbon source, nitrogen source, hydrogen, oxygen, then a calcium, magnesium, or any minerals we will add on it. So I will take 5 variables for better understanding. It is regarded as A, B, C, D, E, F, and G, right? Out of these 7 variables, what I am doing is I am taking 2 as a dummy variable, so which no factor is assigned. Why we are taking a 2 dummy variable? So in this I have assigned E and G as a dummy variable. So the reason why we are incorporating this dummy variable to check if there is any experimental error are there or not. If there is an experimental error, so that we can mathematically nullify with the other original variables. This we will understand little bit detail while we are performing the calculation. So what I have done is I have taken 7 variables A to G. Then I have taken 2 levels, that is an high and another one is the low, L, H and L. So then I am designing a Plackett-Burman design of this all the variables. So you can see in the figure. So I have total 8 experiments only for 7 variables, which by this only we can optimize it. So if you see A, B, C, D, E, F, G is the variable and 1 to 8 is the number of experiments. So in this I have taken 2 levels H and L. Now my work is to allot this high and low for each variable. As on total, the number of high variables should be equal to the number of low variable. How I can do it? It's a simple one. So what I am doing is I am taking a H high in for variable A means I will take another variable. In this case I take example as a B. If my variable A is high, then 2 of the variable in B will also be high. So overall what I will get out of experiments, 2 variable will be high both for A and B. Similarly, 2 variables will be low for both A and B. So out of 8 experiments, 2 will be high for A and B and 2 will be low for A and B. Similarly, if we calculate for A and C, the same case also exists. So I take an example for C and D. You can see 2 high for the C and D and 2 low for the C and D, right? Similarly, what happens is that if I take A variable high, then 2 B variable will also be high. So in this you can see the experiment 4 and the experiment 6, A variable high and B variable also high. B variable will be low. Whereas if A variable is low, then B variable will be high in 2 cases. So out of these 8 experiments, in 2 cases A variable is high means B variable also high. Another 2 cases A variable is low means B variable also low. Then another 2 cases A variable high means B variable will be low. In another 2 cases if A variable is low, then B variable will be high. So total I can end up with 8 experiments. It apply for all the variable. You can compare A with C, A with D, A with E, or B with C, or C with A. Anything you can put it, it will be the same. So first we will do it. Then what we are doing is we will run the experiments, we will get the readings, then we started performing the calculation. So we will perform the calculation irrespective whether it's an original variable or dummy variable. We will perform the calculation in the same manner. Then we will end up with results. So in the results, the first objective is that we have to find out if there is any any interaction is happening between the variables or any problem method of analyzing the response has happened. This can be checked easily by getting the results of this dummy variable. If the dummy variable is 0, then there is no interaction or no problem in measuring the response. If the dummy variable is not equal to 0, then there is a small experimental error or some experimental error is happening. So because of the method of detection of the product or any method of analyzing it may be there. So but this, in spite of that, even though we find that experimental error, still we can counter it with by analyzing the data. So next stage is how we are analyzing the data. So in the first step, what we are doing is out of these 8 experiments for variable A, 4 experiment will be high and 4 will be low. So I am getting average out of this high and average of this low and calculating its difference. That is in the step 1. In step 2, what I am doing is that I am calculating the mean square of each variable. So how we are going to go about it, let us see. In step 1, so the 4 high will be summed up and divided by 4, I will get the average. Similarly, the 4 low, we will get the reading, so we will divide by 4, we will get the things. Then this difference will be calculated by a simple formula, sigma of variable A high minus sigma of variable A low. Similarly, we will calculate the mean square, that is a variation effect. How we can calculate it is the simple mathematical formula. So consider for variable A, sigma of A high minus sigma of A low, that is the difference, whole the square divided by number of experiments. So in this case I have taken 8 experiments, so I will divide by 8. So now I have calculated it, we will get a calculated values. So in the figure you can see for all the 7 variables we get the difference. So for example for A, so that the sigma h is 3.9 and sigma l is 14.9, so the difference is 11. So its mean square is 11 minus 11 square divided by 8, so I will get 15.125. Similarly, I will get for B, C, D, E, F and G. So now what's the problem is that the dummy variables E and G doesn't reach 0. But it is not that much high, so that our experiment is not precise, but it is in negative sign. So we have to calculate the experimental error. So in step 3 what we are doing is we are calculating the experimental error by averaging the mean square of this dummy effect. So we will end up with a reading called 0.0325. So how we got this experimental error is adding 0.045 plus 0.020 divided by 2, so we got 0.0325. So what we have to do is we have to nullify this experimental error with the other variables. This we are doing in the step 4. So what we are doing is, so we have to identify the variable which is having more effect and the variable which is having less effect. So this we can calculate by F test. So how the F test is performed is a simple mathematical analysis. factor mean square of each variable by error mean square. So if you see the values of each variable, for variable A, the mean square is 15.125. I am dividing with the error value, that is 0.0325. I will end up with a value. Similarly, I will perform for B, C, D and F. But I will miss out E and G because that is used for calculation of the error mean square. So out of this, if we see the values, what we end up is that A, B and F, the values are very high enough. So they are significant factor which is need for the product formation. Whereas the value of C and D is very low, which is very insignificant in nature. So our work is to optimize only A, B and F variables. So this can be done with the other methods. So our optimizing level, which one we have to do is, we have to select the major variable which is significant in the product formation. So that only should be optimized. This can be optimized by the two methods, that is Box-Wilson method and the Hendrix method. So how we are calculating with the Box-Wilson method is that it is done by a simple counter plot method. It is in the Box-Wilson method we call as a response surface plot. So in this what we are doing is we are plotting a graph between x and y axis by using the experimental variables. We will take two variables. So we get response. That response will be plotted. So that gives a response surface curve. So if you see how we are constructing this plot is that, so we are conducting a series of experiment by different combination of two variables. So we take two variables, one in x-axis, one in y-axis. We take different combinations, then we get the response point. Then we will delineate it to get the optimized level. So how we are doing it, I have shown in a picture. So if you see, we are taking two variables, x1 and x2. So first what we are doing is, we are taking the x1 fixed, we are changing the x2. So we get a values. Similarly, once we optimized, which is the maximum yield we are getting, there the x1 becomes a optimum level. Then we are changing the x2, right? So sorry, we are getting the optimized x2, then we are changing the x1. So both the things will cross at a particular point. That is the optimized level of both x1 and x2 variable. So in the second method, Hendrix method, it is little bit very simple and easy method compared to Box-Wilson method for optimizing these two variables. So in this thing, so how it is the experiment is started is, we are taking two variables and perform initial experiments, say 4 or 5 experiments. Otherwise, by our previous experiments, the knowledge what we have got it, based on this we can select the particular levels, variables for this variable, levels for this variable, and we can perform experiments. So how it is done? Let us understand the Hendrix strategy. So this is how it is done. First we are keeping a large window for optimizing the two variable. Then we are shrinking the window so until we get an optimized level by an incremental fashion. So let us consider. First we are taking two variables x and y. Then we are designing a 5 experiments of different combination of these two variable x and y. Right? Let's consider x variable is 0 and y is 100. X is 20, y is 80. Then 40, 60 in different increment I have kept it. Run the experiment. What I will get is, I will get an optimum maximum yield of the product. Right? So once I get a maximum yield of the product, then I am creating a new space. For example, I am getting a maximum yield of the product is 30, 70. 30 of x and 70 of y. Then I am creating a new window of performing next set of experiments. So what I am doing is 32, 68, 34, 66, like that I am reducing the values. So in this new space, I will perform another 5 experiments. Then out of this, what I will end up, I will get up here one particular experiment which is an optimized one. Consider 35, 75. Then further I can run experiments until I will end up with that optimized level. So this I have shown with a simple example. A counter plot was performed between two nutrients, that is the cellulose and the soybean meal in candidine formation. So if you see, once optimizing, we are shrinking the window. So until I am getting the optimized level. So this is all about the optimization method with this technique. So you can understand better in my future lectures of other optimization technique, especially the simplex method of optimization. So thank you very much.